All right, hey, how's it going? Coming back at you today. We're going to talk a little bit more detail about the sliding filament theory of muscle contraction. I'm going to flip my screen over here for you guys quick. All right. So yesterday we talked about what happens where the nerve cell connects up with the muscle cell, and it doesn't connect up. There's a small space in between called the synapse, and that's where the neurotransmitter acetylcholine makes its way across the synapse and attaches to the muscle cell, opening up channels that allow sodium to pump into the cell. So the last thing we talked about yesterday was that after that sodium rushes into the muscle cell, what's created is called an action potential. An action potential is the initial stages of the muscle contraction, and that's what we'll be talking about today. So a couple of things right out of the gate here with the notes. So this is called the sliding filament theory of muscle contraction. Activation by the nerve causes myosin heads. Remember, myosin is the thick filament, to attach to the binding sites on the thin filament actin. The myosin heads then bind to the next site, to the thin filament, and pull them towards the center of the sarcomere. Remember, the sarcomere is the distance between the two Z discs. And that ratcheting action of the myosin heads are what pull together the Z discs, shrinking the sarcomere or shortening it for what we call muscle contraction. So again, here's a picture where you can see that. Here's our Z disc. Here, the myosin is the red thick filament. The actin is the blue thin filament. When the sliding filament theory of muscle contraction occurs, the Z disc shorten, contracting the muscle. We're going to zoom in here now and look at the actual myosin and actin filaments. So you can see in a relaxed muscle, the myosin and the actin are not in contact with each other at all. It's only when an action potential sweeps through the sarcolemma, excites the muscle cell, and calcium ions are then released from the storage sacs within the sarcolemma. Calcium is the big thing here that then binds onto actin, and it actually removes an area called tropomyosin out of the way, and it allows the attachment sites right here to be exposed so that myosin can grab a hold of it, okay? And what's really cool, the myosin molecule, it's almost like these little golf club shaped heads. What they do is they act upon that thin filament and almost kind of ratchet it up together, okay? So once the myosin heads are attached then to the actin, ATP comes in. And remember, ATP, adenosine triphosphate, is the ultimate energy source for all of our cells. And when ATP releases its third phosphate bond and releases energy, the myosin heads ratchet. And this happens over and over again to shorten up that muscle, okay? So there's a lot of things happening here, okay? Take it back in order. One more time. I'll just scroll back here. In a relaxed muscle, you can see there's no contact between the myosin and the actin. When an action potential is made within the muscle cell, calcium rushes in from the sarcoplasmic reticulum to the sarcolemma. Calcium attaches to this purple thing right here, and here they just call it a protein complex. Its real name is tropomyosin. When calcium attaches onto that, it moves it out of the way, and it allows the heads to attach to actin. Now, ATP comes in, and it ratchets up the myosin heads to pull the actin together. And it's pulling the actin together that shortens up the actual muscle. So really, it's a pretty complex process if you really go back to what we talked about yesterday and all the things that happen in the nerve cell, then across the synapse between the nerve cell and the muscle cell. And then once that action potential is felt within the muscle cell, it has to be, all these other things have to come in place with the calcium rushing in and the myosin attaching to the actin and ATP allowing myosin to ratchet those together. And I think that this happens in milliseconds. If we had a chance to meet face to face and do a lab together, we'd probably hook all of you up to a EKG, and you'd be able to see the actual movement of ions across your muscle membranes as we would try eating different food. But maybe we'll have to bring in some volunteers for help next week, some other teachers that are in the building, and hook them up to the EKG. If you guys have anybody you'd like to see on the EKG, let me know. All right, I hope this helps. Good luck.